Good morning, everyone. I also wanted to mention that Dr. Garson's involved with some very important CMS initiatives. I think the Center for Innovation or something at CMS. So he has both an academic as well as a very important assisting government role. So we're looking forward to his visit. This morning's lecture, we're delighted to have join us from Rice University. Professor McDevitt is the Brown Weiss Professor of Chemistry and Bioengineering at Rice. He's pioneered the development of integrated bio-nanochips, which we'll hear about during his presentation, in which he takes the advantages of nanotechnology, places them in a context of a molecular sensor with microchip-based technologies to try to create devices that can assess risk, assess early disease states across a variety of different diseases, and we may hear that in his presentation as well. He's a very distinguished inventor and investigator, supported by NIH, Bill and Melinda Gates Foundation, Welch Foundation, Duke Charitable Foundation, as well as the NSF and the Office of Naval Research. He's written or co-authored more than 170 peer-reviewed scientific manuscripts. He holds more than 150 patents or patent applications, making him one of the largest giving him one of the largest patent portfolios in the history of the University of Texas, Austin, where he was previously before joining Rice faculty, I believe in 2009. He's been cited with numerous awards. I won't review them all, but clearly a very distinguished investigator. And what I really enjoyed last evening over dinner with him and several of the other faculty was his commitment to not only innovate and create new science, but to see his science translated. And that's one of the reasons why he was so gracious to accept our invitation here. We're hoping that he'll forge, he's already got a number of relationships with clinical investigators here. We're hoping to forge ties between his program at Rice as well as the Division of Cancer Prevention and Population Sciences here. So thank you all for coming. I think we've got a real treat in store. Dr. McDevitt.
Ernie, thank you so much for that very kind introduction. It's really a pleasure to come speak with you this morning. Let me begin my talk today with a pragmatic issue and a discussion of the healthcare cost. We've all witnessed basically a very challenging situation that our nation is dealing with now. And there's been little thought to the win-win situation, which has been unfortunate. And we are dealing with an incredible problem relative to the cost of healthcare. Going back to 1970, the per capita healthcare cost was about $350. And now it's over $7,500, again, per person. And if we look at where we are in the richest country on the planet and extrapolate how this might become more difficult in a global sense and go on to Africa, where there is 1% of the economic engine to drive healthcare, we find that we have a big problem. Healthcare is nearly pushing the richest country on the planet into bankruptcy. How do we make this process more efficient? And this is an incredible challenge for us all. And so we look at microelectronics, which is an area which I come from. And I grew up in Silicon Valley, went to Austin, Texas, described as Silicon Hills. And so the integrated circuit has been with me for some time. But if you look at the cost structure of the integrated circuit, you find incredible opportunities here with 48% cost reductions each and every year. And we know our next iPhone is going to be cheaper and it's going to be more powerful. But yet our expectations are that healthcare will be 7 to 8% more expensive next year. And new technologies coming into healthcare don't necessarily make your job easier. It often makes it more complicated. And so we need to think about efficiencies in going forward. And today, most diseases are simply diagnosed too late when they're too expensive. And the diagnostic screens have potential to capture people early on. And if you want to do this, you need to look behind the door. You need to look at biomarkers that are useful to capture the disease symptoms in a preclinical stage. And that becomes a challenge. We have lots of biomarkers now. We have very few approved biomarkers. And that's, I think, one of the significant challenges in this area. So we are now witnessing an incredibly exciting time of science and engineering and also in medical research where we have this omics revolution, genomics, proteomics, glycomics, metabolomics, providing us with this powerful information about the molecules of life. And so that's all great, but the brutal reality is that there's 20,000 cancer biomarker papers, and there's 6,000 in cardiac. And yet the FDA approves one biomarker per year for every disease on the planet. One protein biomarker every year. In fact, it's a little bit less than that, about 0.8 biomarkers for the last decade. So we are filling up journals, but we're not creating the translation in the full sense. And this is one of the first rules that I learned in coming to the Texas Medical Center in July 1st of 2009. Soon after that, I spoke with your colleague, Bob Bast. And Bob said, if it's not approved, I can't use it. And so if it goes to the paper and it doesn't get widespread approval, then it's a challenge. And so that really focused our attention on what we need to do, and it changed our scientific activities to focus more on that. So let's imagine a more positive future, basically where we create a bridge between MD Anderson and new microchip technology. And this microchip technology helps us to go global in the same way that an iPhone could go global, and it can go into Africa and leapfrog the technologies that they have there today. But if we created global solutions, we could create systems that could be in MD Anderson, but they also could be in the pharmacy, and they could serve as an additional screening modality. They could be in the dentist office, which is the most frequent medical contact that we have, on the order of twice a year. They could be in people's homes, and they could be in the hut in Africa. And so this is really a pipe dream today. But in my talk, I'm going to basically give you a progress report about where we are in achieving this vision. And this comes at the cusp of us just receiving a large grant from the CPRIT, from the Texas Cancer Prevention Research Institute of Texas, which has allowed us to establish the Texas Cancer Diagnostics Pipeline. So this is part of one modality, one disease class that we're moving this new technology into. So I've mentioned a little bit about the problems with the current diagnostic infrastructure. How I'm going to organize my talk is shown here. I'm basically going to tell you about our bio-nanochip approach, which creates a sensor that can learn. And that's a key thing, a flexible sensor that has something like human intelligence. You give it more input, and it makes a better decision tomorrow. In order to do this, we need to select biomarkers carefully, and this is a huge challenge right now. And so I'll tell you how we're doing that now, and then bring a couple of case studies so that you can see where we are midstream en route to building what I'd like to describe as the biomarker highway. So Texas has allowed us to, in this state, begin to play a leadership role. And I think MD Anderson has already established this leadership role, but it allows additional augmentation of efforts, and to have the state of Texas now provide a funding base for the next decade, $300 million, that really can accelerate the process. And so we're very excited about participating in this. And we've put 3 cancers into our diagnostics pipeline, and I'll tell you about them later on the talk. But before I do that, I need to give you Biochip 101, which gives you a sense of how we put this system together. Now, we have a very good team at Rice University that you see up here. At this point, I'm very pleased to have these coworkers, but also very pleased to have what I would describe as the dream team of clinicians. It's been literally our dream to come here and be your neighbors and to be able to help in the next step in the healthcare, to make healthcare more efficient. So think of us as your servant. This is not about us, it's more about you. And we want to empower you to have the next generation of tools to diagnose cancer and to monitor cancer in a more efficient way. And so again, my job today is to inform you about what we have, and then maybe we can brainstorm about how we get there together to generate the next series of tools. So let me make the analogy to the software industry, to the information highway, which we have today. And this is such an incredibly powerful tool when you think about it. When's the last time you went to the Yellow Pages and looked up a phone number? It's completely changed the way we gather information. Scientific and clinical articles, same thing. We get access to that. We have information at our fingertips and search engines that are incredibly powerful. Now, in the omics area, we have journals. And although that's powerful information, again, we have a disconnect. These are not FDA approved, and so we're down to this one biomarker a year, and we have a knowledge gap. And that knowledge gap is getting in the way. And so basically what we want to do is we want to create something that might be a game changer in this area. So it's the iPhone applied to diagnostic and screening applications so that information flows when you need it. And if you had that, imagine what microelectronics has already done. It is something that's predictable, and it's something where you can define your next set of goals. We have Moore's Law, and that tells the semiconductor industry 18 months from now, the feature size is going to be a factor of 2 smaller, and your computers can be twice as fast and it's going to cost half as much. Now, that same tool also creates the next integrated circuit. So if we can, and that's allowed this area to grow virally, if we could create a tool that also gathers information in the biomarker space, then that empowers the next generation of tools. So I want to create this ambitious goal. It's not here yet today, but we're beginning to see the fruits of this concept by virtue of dealing with one disease, creating a line of code that's then applied to the next disease. And again, that'll become more clear as I show you where we are. So if you look at the inefficiency which we have today in terms of approving biomarkers, and this is one of the first things that we did coming here to the Texas Medical Centers. We established the Gulf Coast Consortium on Early Disease Detection. And we had a series of meetings which we analyzed the approval process and identified a number of linkages that were not made. And so they are related to things like having 3 different technologies, let's say a mass spec to gather discovery information, then an ELISA plate done by a second group to validate that, then an immunochromatographic or home pregnancy kind of test being the final implementation. So 3 different technologies, 3 different sets of interest, and a very inefficient pathway from this to this next step. And so that's a huge dead end, a huge barrier, and it's something that limits venture capital in the real world from coming in and taking our discoveries and bringing them to the next level. So there is a technology gap here, and we need to bridge that gap. And so 14 years ago, we began to look at a new way to do chemical sensing. And at that time, I had no idea I would be here today talking to the best clinicians in the world in cancer. We were focusing on Coke and Pepsi and creating an electronic taste chip, believe it or not. And that particular taste chip, though, with time, became a rich source of information. Instead of doing salt, sweet, sour, and bitter, multimodal testing, many classes of chemistry, but a smart algorithm behind the back of that. So that was the concept of this electronic taste chip. We began to apply it to clinical applications, which I think are far more helpful to society. So this particular individual eating, let's say, a strawberry here has some kind of pattern recognition in the brain that's happening that brings back a whole series of emotional responses about that earlier lifetime experience of eating that strawberry that tasted good and you didn't get sick to your stomach afterwards. And so we gather information during a lifetime. As an infant, we know sweet is good, we know bitter is bad, and that's about it. And everything else we learn. So our chip is built on that same concepts, many chemistries that learn. And so the brain or algorithms is an important part of this. But just like the brain that has a left hemisphere and a right hemisphere, we have two classes of sensors. So I'd like you to latch on to that because that's an important part of our capacity to create a universal sensor system is a soluble chemistry system. And we describe this as a chemical processing unit, the left hemisphere. And then we have a cellular processing unit which captures cells and spores and bioparticles in general, and that's the right hemisphere. So we have two CPUs. Now the CPUs come together and work within customized chips that go into an analyzer. So that's the other part of this. And the analyzer is basically a box that is a souped-up digital camera. So we leverage the digital photography industry in doing this, and ultimately we're going to snap pictures. And we need to do something on the chip is collect proteins or collect cells, stain them in a specific way, and then snap a picture. And then software goes in and it interprets that. But when we do this, go through the image-based approach, what we're able to do is program the chemistry into a variety of different areas, which are more traditionally done on refrigerator-sized instruments. And having a common modality means that we can have this portable box go into lots of different areas. So let me just show you a little movie sequence about the first application that is moving into widespread clinical practice. And this is HIV immune function test. I won't spend a lot of time talking about this today, but just to give you a sense about how we process the sample. You saw a drop of whole blood go into the chip, and that's 99.9% erythrocytes. We got to get rid of them. And that happens in the cellular processing unit, and we keep behind the more rigid white blood cells. We don't see them yet, but we capture them. And the next step is a little bit of chemistry. So we capture the cells, and then we stain them with the same types of reagents which are used in flow cytometry. But we get away from the flow cytometer, and we snap pictures. And so this shows the green channel that the analyzer is collecting, and this happens after a CD4 antibody labeled to Alexaflor 488, so it spray paints the cells green, that cell population that carries the CD4 epitope. And then we bring in CD3 antibody in red. And if you remember what happens in flow cytometry in this particular area for counting CD4 positive T lymphocytes, it's the overlap of both of them. And so we have a digital process to do this, and we do something that's incredibly simple. We count yellow cells that are around, or yellow objects that are around in this big, and interpret that as an 8-micron cell, and that has very, very strong accuracy to correlate with a flow cytometry. So the miniaturized sensor that can go in the hut in Africa is taking that same kind of measurement that's done in this very expensive instrument and reducing it to something that's analogous to what is done for blood glucose measurement. So it's that kind of simplicity. So I think when you look to the implications here, the tests that you may consider complicated today that are off limits for you to do point-of-care testing today, that's not going to be true tomorrow. And so this is one of our challenges, I think, as we get into interactions with new clinical partners, is we need to think past today's limitations and begin to dream what would be possible tomorrow, because this is something that I think allows us more options. So in the CD4 case, for HIV immune function, we have a chip that looks like this, and our cancer chips look like this, and our cardiac chips look like this. There are different reagents on the back. They're always a credit card, but one of the keys here is what we're replacing. There's no pipetting step. So we're not trying to add additional burden to the clinician. We're trying to make this a glucometer. And the phlebotomist doesn't have to be there either. It's a drop of blood or a couple drops of saliva that are self-metered in this chip. We do not need stable power, maybe less of an issue for you, but very important in the Africa setting. We do not need the cold chain, and we don't need the flow cytometer. So these are all systems that are an infrastructure changing element that happens by virtue of having this kind of new approach. Now, a little bit more about how we pull the signals out. And there's two different pathways because we have a left hemisphere and a right hemisphere. And if we look at the cells at the surface layer, they look something like this. At lower magnification, let's say this is a CD4T lymphocyte, we have 10,000 copies of CD4 on that cell. We light them up like a little bitty Christmas tree here. This is one individual cell that has many different copies, but we see it as one red blob. And then that comes down, it's measured at the north pole, basically, of this chip. And then this goes into the analyzer, and this digitizes chemistry and biology. And so this information can flow over the internet, and I think that's more important in Africa, where we'd like to do disease geographical topo mapping, but I think that can lead to additional efficiencies in the US too. So that's for the cells, and for the proteins, we capture things at a bead array, and each bead is different, and each bead is a different test. And so, again, this is all based on an imaging-based approach, and ultimately what this allows us to do is to think about all the different instruments that exist today, like the flow cytometer, and the assistant to a pathologist. So we don't eliminate the pathologist, obviously, but what we do here is we provide a tool in, let's say, oral cancer, so that you don't have to do surgery, grab a sample, and then process it for 3 days, and then look under a microscope with an expert to provide that diagnostic. We have a fast way to do that, and I'll come back to this oral cancer example in a minute. Clinical analyzer, and then this LC mass spec mass spec unit is an example of something that we're doing in the addictions area for with Tom Costin at Baylor to develop a chip that looks at roadside drug testing. So these are all analyzers that are gold standards that are basically our target to try to create a strong correlation, because we want to build an instrument that's miniaturized but is as good, if not better, than that larger instrument today. And this is a key issue that hasn't happened today yet. When you think point of care, you think of two things, probably. You think of that it's more expensive. Yeah, it's convenient to be next to the patient, but it's more expensive and it's a compromise relative to the lab instrument. So it's inferior in those two ways. That doesn't have to be the case going forward. Again, microelectronics has done that. We don't think about going back to the abacus now. Microelectronics is the best way to do these kinds of calculations. And so just to give you some kind of sense how we could both miniaturize and be better, this gives you a sense of our bead array that is built on these 200 micron diameter agarose beads. So this is basically derived from seaweed, the same bead that's used in affinity chromatography we use to capture proteins. And that has a very high surface area, and we've engineered this to have a nano net, and it's a micro sponge. And all these characteristics allow us to get reagents in very quickly. And it has high surface area, and ultimately this makes this a better instrument than the gold standard. And that's borne out when you look at an ELISA plate, let's say for doing a gold standard measurement of a protein, it builds up a signal from a surface. And if you remember your chemistry in Beer's law, the amount of signal you get is traceable ultimately back to the thickness of your sample. There's not much sample thickness here, so we have to enzyme amplify, and it becomes a slower process and less efficient. So our miniaturized sensor now is outperforming the refrigerator. And this is borne out in these published studies in the area of CRP, where there are 13 FDA-approved CRP tests now, and we lower the limit of detection by a factor of 10 to the third relative to the best instrument out there today. And so that empowers us to take the next step and to have credibility to get into clinical trials, of which we are in 6 now. And they are summarized here in the areas of oral cancer, cardiac heart disease, drugs of abuse, and CPRIT has allowed us to put 3 things into the pipeline: low prevalence or oral cancer, ovarian cancer, and prostate cancer. And so I think we're at the beginning of this pipeline, but the pipeline, although maybe a little leaky right now, it's being established. I'm sorry, Shine. CRP is an acute phase reactant, C-reactive protein that is used in lots of different areas to look at general illnesses. There's a high sensitivity CRP test that's also used to look at risk assessment for future cardiovascular disease, which is actually better than cholesterol now in making that call. Now, sampling, and Shine, thank you for that question, by the way. Sampling is an important issue because we need to think about this. If we really want to be point of care, then we need to have this full process thought through. And all of our trials have this as a consideration now, a non-invasive sampling modalities. So instead of vein puncture or venipuncture, it's a drop of blood. Or in the case of cardiac heart disease, it's a gum swab. Or in oral cancer, it's a brush biopsy instead of surgery, doing either a punch biopsy or a scalpel biopsy. So these elements now are bringing together the pieces that allow us to start dreaming a little bit further ahead. And so the biomarker highway, which I've alluded to today earlier, doesn't exist, but we can think about building an infrastructure that would scale. And so how this would happen would be to have chips involved in the discovery, in the validation, and in the actual clinical implementation. And in all of our trials, that's what we're doing. And that's a key factor, we feel, to make the process, the pathway to the FDA, to be more efficient. So this is a tall order, and the construction of the biomarker highway is still happening. So we have the construction sign next to it. But let me show you some of the pieces which is accelerating the process. The first is a computer that does a computational fluid dynamics thing. We're at the point now where once we know which antibody and which protein we need to detect, we can model that. We can build a chip. So I've just shown you subtle changes in how the protein is captured at these beads. This agrees very well with what we see in the lab. And this is key because microelectronics already does this. They're computer tools that design the chips, and that removes the slow process. Now, the other thing we have to do is we have to build an infrastructure to make these chips, and we do this with microfabrication. And so maybe this part is obvious that this is already happening in the semiconductor industry. Less obvious is that we could do this in the medical space, but that is indeed what's happening now. We also need to make sure that the sampling scales, and so this needs to work for you, and this can work for you if we have something that human beings can handle. And so this is a credit card. Even though our bio-nano chip, again at the north pole of the chip, is so small you can't even see it. So we need to use electron microscopes to image what's happening there. But you need to have the clinical interface, the macro to micro interface. And that's happening in this kind of modality. You see a couple gray things here too. These are the blister packs. So all fluids and all reagents are on the card. The only thing that you add is the sample. And then everything else happens in the analyzer. Now the chemistry is mass produced too. And again, we didn't do this overnight. This is a 14-year exercise. These are our lines of code though. And we are making 1 million to 1 billion copies of little chemical microreactors at a time in this little vessel. And then we pick and place them into the chip. And so that's our line of code. And that process of picking and placing isn't so bad. And then the next part of the highway here is making the bridge into the assay, because we need to do assays. And we do this with the help of robots that begin to automate this, this process. Now, I mentioned the sensor that learns, and so we are in school now. And MD Anderson and the Texas Medical Center is our classroom. And these systems are learning, and my new home is shown here in the Bioscience Research Collaborative, which is a $300 million investment by Rice University to be away from the core of the campus and to be over here. And we really want to create the bridge over here. And so let me now switch gears a little bit and move into the clinical implementation of this. And in first area will be cardiac heart disease. And I understand cardiac heart disease is maybe not your main theme unless you're treating breast cancer patients with anthracycline and you have severe cardiac toxicity. But we have a large trial now with Baylor, and we have been involved in selecting biomarkers and moving them to an oral test. Now, just a quick statement on what we did here. We went to the literature, found 3,000 proteins, and then down selected to those 177 that are used in the diagnostic and space here, as well as in the papers. And then we, with PubMed, went down to 32. And then we started the experiments. And then we found the top 5 through a variety of different themes. So we have a pilot study and then a validation study. So we're going through these 2 steps. And this is key for everything that we do, is we need the extreme phenotype initially. We need the softball pitch initially so we can make our chip so it begins to learn, it gets into the right space. We get comfortable, we build the muscle mass, and then we move to the next blinded study, prospective kind of trial. So we're going through this. And we did this to find the biomarkers that are now in our cardiac chip. Now, the reason for me spending a little time here is just to emphasize that when we stop here and look at which biomarkers work, and this is what this particular slide shows, the blue curves are the diagnostic, a feeling of the diagnostic information content coming from salivary biomarkers that had never been used before. And our chip was able to pick them out because it was ultra-sensitive. But when we analyzed all those biomarkers and went back to the mass spec experiment, we found less than 1/3 were actually in our final panel. So the vast majority of things moving from mass spec to ELISA onto immunochromatographic test were not giving us the information content. The biomarkers that work are not found by mass spec in this case. And that taught us that, not that we're going to get rid of the mass spec, we need that. That guided us, but it taught us that earlier we get the discovery tool put into place, the better. And that means if we're not, then we're being inefficient at the next step. So this is now moving into a large trial with Christy Ballantine and Bikem Bhaskar, your neighbor at Baylor College of Medicine, also our neighbor too. And it's allowing to create this kind of chip. And so this gives you some sense of what we see in a real-world chest pain patient. False alarm, part of the 80% up here, doesn't light up. And these strategic proteins that we found through that sieving process. Authentic heart attack over here is lighting up like that Christmas tree. And we have calibrator beads, we have negative controls, a number of different things that you'd like to do to make this a valid measurement. Now, another thing that we learned from this particular trial is that we can not only think about the first diagnostic indication. With the chip, we can load it up to the second trial and the third trial today. And we are doing this in this trial. And again, it's a type of adaptive clinical trial that's future looking. And so where this has implications in heart disease is, instead of focusing on the last minute of someone's life, which is the goal of our first trial, the AMI diagnosis, we are looking at all 7 different phases of the cardiovascular cascade. And these are the biomarkers here that are in our chip. We don't need all of them to diagnose AMI, but we gathered that so that in the 80% of the population that's not having the heart attack, we can follow them up and then look at their risk for a future heart attack. And we can also look at congestive heart failure for that population group. And so Christy Ballantine is in the risk space and Bikem Bhaskar in the congestive heart failure. And we have all of that covered within this single trial. And I think that's a big deal. We're very excited to take those next steps now. Now, let me get back more to your background in the area of cancer. We have 2 trials moving forward in oral cancer. And it began with the same kind of process that I've already alluded to. Extreme phenotype, softball pitch first, which is what we did here, and we distinguish between the normal patient and the oral squamous cell carcinoma patient. And we created a chip that's an adaptation. We reprogrammed our HIV chip. But we did this with a different set of reagents. And now we have a 3-reagent cocktail that comes in, and it does red, green, blue. And red, green, blue are hitting these cells, and it becomes something analogous to immunohistochemistry, but it's fully automated. It's here, it's now. And you see the sequence of what the analyzer does is it snaps 3 different pictures at different focal heights. So we have a suspicious lesion, we collect cells, we introduce them with a buffer into the chip, and then we get clumps of cells, and we have to deal with that. They're not all at the same focal plane, so we snap 3 different pictures. And then we stitch them back together into one focused image. So it's like confocal or deconvolution microscopy. And then we profile the cells. We find the nucleus. We find the cytoplasm. We pull out a whole bunch of stuff, 48 different imaging parameters. And then we ask the question, what's the best way to make this diagnosis? And we read the literature, and the literature says 80% of oral cancer patients have EGFR overexpressed. So we put that as a surface marker. And we find that that's decent. But we went into this process thinking that would be the best. But we do a logistic regression area underneath the receiver operator characteristics curve to look at the diagnostic input from these different variables. So that's what's happening here. Again, EGFR okay, but much better for the nuclear to cytoplasm ratio. And even better yet is the combination of the EGFR signal and the nuclear to cytoplasm ratio, and that gives us the best result down here. So it's a model, an image-based model that allows us now to think about the next step. And we know by talking to you and your colleagues that you're really interested in the dysplasia transformation into cancer. And so this has become the focus of our next 2 trials, is that transformation. And no problem, we have a chip, it's flexible. We look into the immunohistochemistry literature and talk to our oral surgeon colleagues in this space, and we add 6 more biomarkers that have a precedent for providing that point of differentiation. So that's what we're doing with 3,000 patients now. And 6 months from now, maybe 3 months from now, I'll be able to tell you the best combinations, but that's actively where we're at. Now the second biomarker that's in CPRED is in this particular space, a prostate cancer. And this is the most common cancer for men outside of skin cancer. And we're working with Ian Thompson in San Antonio to help push this forward. And in this particular area, we're looking at what is now a C-grade test, which gathers about 70% of the male population. And if I was teaching a class, that would get a C. But there are additional things that are known today that can bring that up to an A grade. And that involves some race information, age information, prior test history, and a combination of a couple biomarkers which aren't bundled. So the limitation now is bringing together all this information at the point of diagnostic. So this is why it doesn't make sense to send this off to a lab and react later, but to have everything there at that point of diagnosis. And so our chip now is doing free PSA, complex PSA, total PSA, and bringing in the prostate calculator all into the same setting. Sorry about that distraction. So what this allows us to do is think about a new kind of testing. So there may not be a regulatory pathway for this today. So I'm projecting into the future a little bit, but this is a modality that Ian Thompson suggested, that we look at an automatic reflex testing system where we do PSA first, that's approved, and if it's at an extreme, that's where you stop. You do surgery if it's way, or move on to the next step if it's very high on the top, and then bless the patient as being healthy if it's very low. But the intermediate result is a problem, and that intermediate result can reflex into the next stage, and that reflex test would basically activate the chip, which already has made this measurement, and then report that, so you could do this in that same setting. Now, with your colleague, Bob Bast, in ovarian cancer, we've begun to develop a point of service test, again working with a drop of blood. And with Bob, it wasn't clear initially why you would want this to be point of service. And so that's another interesting thing that we learned. But with time, it became clear that if this test could be done in combination with a transvaginal ultrasound, that a CA-125 and an augmented test in that kind of setting has diagnostic utility. But there you would need to reduce and gather information in a single visit. And so the point of service test makes sense in this kind of environment also. So we need to be very good in our analytical characteristics here because this is a relatively low prevalence disease. And so these are some of the requirements that we're looking at: high accuracy, good reproducibility, low limit of detection, and multiplexing. This is key. CA-125 is not enough. It's an augmented CA-125 that is going through a number of validation trials that looks interesting. And one of the challenges, if you read the literature in this space, there have been other groups that have tried to bundle together CA-125 and markers like HE4, which are on the top of your interest level, and haven't been able to do that. And so this is one of the first things that we began in the CPRIT effort in this space, is to look at the interference effects, which are explored here between CA-125 and HE4. So CA-125 responds to CA-125, not to HE4, and vice versa. And this has now gone through a quick and dirty validation step. So this is our, again, softball pitch. These are stage 3, stage 4, but very good capacity to distinguish between healthy and diseased. And so that's borne out in these ROC curves. And again, I don't want to claim that what we've done is invented a new panel just yet that's better than what exists today. But what we have is a chip that's now competitive that can make this measurement. And our next step is to look at low prevalence populations and do longitudinal studies. So I'm ready to wrap up my talk here by closing with a couple things. And I'll be happy to share this laundry list, which I'm sure you're not going to be able to memorize right now. But we have 30 different biomarkers we're moving through clinical trials now. These are our lines of code. These are the soluble chemistries. And they are there to begin collaborative things in other diseases. And we have these 6 cell-based tests. And we're not limited to either protein or cells. I think some of the fascinating combinations actually going forward may be combinations of these tests. And cardiovascular disease, we know, has one such example of taking 2 non-specific tests, bringing them together, and creating the best risk assessment strategy that exists today. And so we're at this stage of putting a little bit of cement on what I'll describe as the biomarker highway. It's not fully paved. It's not all the way to the FDA yet, but the FDA is clearly a strategic relationships that we're building, and we want to move these into the next generation of test and ultimately help people. That's our goal. We want to help the patient. We want to help you. We want to begin to help make healthcare more efficient. Well, let me end my talk there, and I really appreciate the opportunity to come by and chat with you today.
Thank you, John. That was wonderful. We definitely have time for questions. I'm going to turn up the lights, but if you need to leave, please feel free to do so.
Thank you, John. One of the questions I have is, have you had any interactions yet with the FDA around, and I do more drug development than device development, so I don't know their approach to this, but typically if you're developing a drug, you meet with them a couple times in the course of it to lay out the developmental plan, at least to begin to interact with them, to define what would be an approvable endpoint, if indeed you make that. Any sorts of those interactions with the FDA yet around these sorts of devices that you know by yourself or other companies?
Right. Well, about 5 years ago, we launched a company in Austin, Texas, whose goal was HIV immune function release in both the U.S. and resource-poor settings. And so they had a series of interactions with the FDA. And we are now in the process of looking at more U.S.-based applications. And I think one of the lessons learned from this experience is if we only focus on resource-poor settings, we're not going to be successful. We need an economic engine to do both. And so I think there's some wonderful synergies between resource-poor settings, creating a trajectory where it drives down U.S. healthcare costs by forcing us to be affordable in that kind of setting. But the FDA trajectory, too, helps to make that kind of testing more reliable. So they're good synergies. But the next steps on all these applications that are in the 6 clinical trials, we're establishing relationships with the FDA right now. There needs to be many more conversations. I think there also has to be an education on both sides because we're looking at sensors that learn, and the FDA gets very nervous about bringing in a new algorithm at the last minute. And so we need to adjust to that. We need to, and that's part of our trials is that we go through a process that typically has 2/3 of the samples devoted to an open measurement, fixing the algorithm, and then we don't touch it, and it's blinded, and third party arbitrates and gathers the data. So we have CROs and funding agencies in that kind of oversight capacity to make sure that we're not tweaking at the last moment. And I think there's always a tendency for the device or the gadget maker to want to do that to get the ultimate results. And so that may have short-term benefit, but the long-term translation is slowed down in that.
Yeah, knowing the FDA's conservatism, it'll be interesting to see how they adapt to a learning device that changes its interpretations with more data.
Can this chip measure the multiple proteins simultaneously?
Yeah, and I'm sorry I didn't emphasize that more, but yes, the multiplexed capacity is a big part of what we do. In every single trial that we're in right now, we're measuring multiple proteins, or for the cell-based test, again, multiple biomarkers or nuclear staining combined with surface staining combined with intercellular staining. So it's key that we get information from different channels, and I think that's another big point of differentiation from a lot of current clinical tests that look for the magic bullet across the entire population. But you can lower the bar with respect to your selection of biomarkers by getting a combination of biomarkers that works better than the single biomarker. And again, we have to be careful about this, and we understand the pitfalls, and there have been pitfalls before with multi-marker test that in many cases only did what I was describing earlier as the softball pitch, only the extreme phenotype, and then changing the population group later on, and then trying to validate that. That if you don't have the same population group during the training as in the validation step, then you're in a very risky position, a very vulnerable position. So it's harder to do it that way, but ultimately I think the success, you're more likely to be successful by doing it that.
Have you applied your chip to differentiate the normal cells and the pre-cancer cells like polyps?
That's what he's doing in head and neck at least.
Yeah, we have some initial information here that's exciting, and so it shows a progressive change in the score, the multi-parameter score going from normal to dysplasia to cancer. And we need to look at this more carefully to say what is the diagnostic accuracy, how well can we stage the different stages of dysplasia. And but I guess I'm hopeful on that. The data will tell the true story, but we have a non-emotional way to report the answer, which is based on the diagnostic accuracy and area underneath the receiver operator characteristics curve. But I guess in this space, and it's one of the reasons why we've chosen this space, is there's not another tool that does this. There's not a non-invasive tool for oral cancer patients. There is a scalpel biopsy, and there's a recommendation after you have dysplasia to come back every 6 months. And people get the surgery the first time, and then they have the sore in their mouth, and they don't come back until it's stage 3 and stage 4. And this is why we're where we're at today, with not much improvement in the outcomes for oral cancer patients over the last 30 years. And so a non-invasive test will allow that another option to provide information to the patient and healthcare providers.
So we're a little bit over time. Maybe we'll just take one or two more. That's it.
Yeah. So can you like like your chips applying for normal cells, tumor cells? Can you also differentiate like the cell type, like the epithelial cells, stromal cells?
Sure. If you can see it under a microscope, then you can imagine doing this in this kind of modality. So we have done 3-part differential and 5-part differential blood typing by doing this in a different way than a hematology analyzer, because we have instead of a Coulter counter or conductivity kind of measurement, we do an image-based measurement. So we bring in a variety of stains to differentiate the different cell types, and stains and surface markers, but that allows us to be in a position to do that. I think we would need to talk exactly about the application that you may have in mind, but if there's a way to do this on a microscope with stains, then that's a good starting point for us to think about how we would do that.
One last question.
From that limited experience with chip-based technologies, I know that on top of expense is an issue, but another issue is cost positive and cost negative. And I'm not sure you have a plan or you have initial evaluation of these issues regarding the chips that you have developed, or you could expect to see like a comparison between other technologies on these issues.
Yeah, no, I think these are great questions. So if you look at other chip-based technologies like the Affymetrix chip or protein arrays, it's different. So it's a different way of doing the measurement. And so a gene chip, for example, which, I don't know, you may pay $600 a chip, and you process that for 3 days, and then you get the 20 SNPs that are 20 sequences that you're going to apply in the next stage. But that's a discovery experiment, and more likely you don't want to spend $600 to measure those 20 SNPs going forward. What you'd like to do is to prioritize and customize something that only measures those 20 and does it in a better way than a gene chip. And in the other areas, so antibody chips and protein chips now are building up a signaling capacity in an area that is now, I don't know, about a decade behind the gene chip in terms of quality control. And the spot-to-spot reproducibility is an issue. And the background response is an issue. And the cross-reactivity between the different reagents is an issue. So these are all problems, and so this is an emerging industry. We face the same problems here, and we spend a lot of time selecting reagents, and we do this on a different kind of platform. We build up signals not from glass slide that has more background interference. We use an agarose bead that is more tolerant to background responses. And I think that the key is in the execution, and it's on these kind of issues that you're pointing to. You need to get good measurements. You need to have good reagents, and we spent 14 years figuring out how to make high-quality measurements now. And we're at the stage in the ovarian cancer area, and I think this is our best case because we knew we had to drive down the assay coefficient of variance to something very low. We're at 3% now, which is very competitive and much better than ELISA, which may be 15%, and the protein chips being even worse than that. So the protein chips are nice because you can do discovery on them, and a commercial supplier provides that to you. But I think these are intermediate tools that once the discovery is made, a better tool is needed to put it into clinical practice.
In terms of oral cancer, you mentioned carcinoma. What about other kinds of oral cancers? Sarcomas and lesions. Have you had experience with any other kinds?
We haven't looked at other types of oral cancer just yet, and I think it'll be interesting to do that. And oral squamous cell carcinoma, being the most prevalent type of oral cancer, was our first step. But I think we now have the infrastructure to more quickly move into that space. And so if you have some ideas there, we'd love to chat.
Well, thank goodness he's only across the street.
Right.
So we are definitely going to have you back. I had 8 questions myself, and I already had to run because he has a meeting. But he had a whole bunch of questions and wanted to follow up with you anyways. So please, the rest of you, please join me in thanking our wonderful speakers.
Thank you, Sean.
Thank you so much. Okay, let's close this down so we can do the finish the Camtasia thing.
Okay, I have to figure out how I do that.
Thank you, John.
Okay, thank
See
you. Thank you for all those questions, by the way.
Okay. I will contact you for more questions.
Okay.
Yeah, I think we're going to have to arrange a meeting for John to come back over and talk to everyone.
There are so much collaborations we can do.
Absolutely. That's what he wants.
No, that's why we came here.
Okay, I need to go.
Okay.
Okay.
So John is the
Okay.
Yeah, we definitely have to.
Let me just shut down
Yeah.
this Camtasia thing. Okay. It's hidden here somewhere. Here.
There you go. Quit? No. Hey, Camtasia ladies.
Do we have Camtasia expertise here?
Yeah, oh, here, stop recording.
Oh,
That would
okay.
be it, right?
Yes, you got it.